are probably important when you're getting your clinical trial application in. And actually, this is also pretty broad. And the idea is really to go out to those who are doing early experiments and people in the labs, not only those that are expert. Clearly, this is not aiming at the experts in cell manufacturing, but it's more on what is the kind of thinking that you need to have in order to get your clinical trial going. And clearly, we're going to focus on cell therapies. And maybe I want to highlight a broad example of cell therapy, and it's basically stem cell transplantation. And for those of you that are not familiar, we're taking patients that are failures to regular treatment and with disease on board, and you have here leukemia cells that are in yellow. And in these patients, we administer high-dose chemotherapy and radiation to kill off the cancer cells. But actually, by doing that, we also eliminate the stem cells, the bone marrow stem cells, and therefore patients are left with only transfusions to get their red blood cells and platelets, and we cannot replace the white blood cells. And we would be waiting very long for these to come back, if ever they are to come back. And in order to replace those cells that are eliminated, the bone marrow cells, then we need to get the source of stem cells. And we can get these cells either from a compatible donor, either a family or a world registry donor, or from somebody else who's family related, who's usually half match and what we call haploidentical, or an unrelated donor. And what I want to highlight with this is that this is a source of cells that we can then take these cells and administer them to the patient to reconstitute their marrow. And this is probably the first example of using cell therapy to treat and cure patients. But actually, there are a large number of applications right now to be using these cells, either unmanipulated, but more importantly, manipulated, modified in order to exert the activity that we want them to do. And then recently, there's the addition of gene therapy, which is now becoming a real issue as this is now authorized in centers all across Canada. Health Canada having authorized two gene modified cell products for the treatment of patients with B-cell acute lymphoblastic leukemia and also lymphoma. So these cells are promised to give a treatment, hope, and therapeutic option for many types of diseases. It's now started with acute leukemia, but soon to follow in disease like heart disease, brain disease, skin disease, where you need actually cells acutely. For example, after a myocardial infarct, where you want the cells to be helping the heart to regenerate and recover good function. The brain, similarly for the brain and skin and burn victims. But there are also long-term needs in other diseases like Parkinson and Alzheimer's and ocular diseases, kidney diseases. Basically, every organ you can think of could be in need of cells. And what's expected to them when you're using cells is that you're addressing an unmet medical need. And if there's something that is needed that I would like everybody to remember is the unmet medical need. When you're going to Health Canada with your clinical trial application, you should have in mind to treat something where clearly there's no curative option out there. And identifying the right objective and the right disease up front is really crucial. And I always emphasize that when you're developing your treatment, aim at identifying the best therapeutic setting to use that. And for that, you need to get your clinicians on board very early, even at the stage when you're working in the lab, because you want to be able to set your cell product in the right conditions that is going to allow you to treat patients in that fashion. So, and with that, you want to have improved the patient's quality of life, or you want to alleviate some of the traditional drawbacks associated with common chemotherapy, not chemo, but any agent, any therapy. As you can see, I'm biased. Being a hematologist, I always think of chemotherapy, but lots of other drugs and agents that have their side effects. Significant increase in patients' life expectancy. Obviously, that's a major plus. And when possible, you want to cure the disease. And clearly, these new technologies are groundbreaking and will be even further groundbreaking in the coming years. So maybe just to highlight that when you're comparing pharmaceutical versus cell-based therapies, there are a number of differentiating issues. And here's a list of advantages. Clearly, I would say maybe it's a little bit biased towards cell therapies, but nevertheless, these are totally true. Like when you're administering drugs, you have to administer frequently. You're injecting a product directly, so therefore you need a highly pure, you need to have pharmacokinetic studies, you need to do toxicology studies, extensive risk for carcinogenicity, and then when you're administering high doses of drugs, you could have side effects, some of which could be severe. And in all these instances, it's usually a limited possibility for cure. And this is not personalized medicine. And therefore, if we are to use these personalized cell therapies, it's possible then to administer cell either as a one-time injection or multiple time injections. And because you're using the agent, you're modifying the cells with agents that are only treating the cells, you have much less chances of having side effects towards the individual, towards the body. And therefore, you're injecting traces of drugs, and you have less pharmacologic issues. Therefore, many times it's not even possible to do pharmacokinetic studies. There's less toxicology issues, although you still have to do toxicology study. There's a risk for carcinogenicity and genotoxicity, but usually because of the low doses, this is much less of a concern. And then you're limiting the adverse effect associated with all the components that are used to expose the cells. And most importantly of all, there's a high probability of cure, and that's why we're all working on developing cell therapy products. So here I've listed a number of products to give you some examples. Clearly, as you can see, they go from the arena of immune disorders, immune effector cells, tissue repair, metabolic replacement, gene replacement, and there are various teams throughout Canada. And Canada is really in a leadership position in terms of cell therapies, and we have wonderful researchers. I'm sure many are on the line right now that are doing groundbreaking work and will bring novel cell therapies into the market. So there are a number of challenges when we are thinking of developing cell-based therapies. And we need to engineer them. There's the raw material. Where are we going to get the cells? And where are we going to get the products to which we are going to be exposing the cells to? So this is very important. And obviously, every component that touches the cell, even the flask, has to be evaluated in terms of efficacy, toxicity, expiration dates, potential for side effects, etc. This may, if you're using cells from autologous, come from autologous or allogeneic and even xenogenic sources, although recently the xenogenic sources have been less popular. Cells may be extensively manipulated, and you see a list of manipulations that can go on from activation to gene modification, encapsulation, etc. And the whole idea is to really limit variability and heterogeneity. There is inherent heterogeneity because of the nature of the cells, like cells are different from one tissue to the other, and they're different from one individual to the other. And therefore, you want to be able to control as much as possible these differences. And you need to have a process that allows you to be validated and to ensure that your process is clearly under control. So maybe a few examples of cells and the advantages and disadvantages. It's only 2 slides. One on ES cells, embryonic stem cells. They're available, they're immortal, they're pluripotent. I don't have to go over that with you. But there are a number of obstacles. These cells are tumorigenic, so there's potential for forming teratomas. And depending on where you're injecting these cells, there could be causing problem. For example, if you're injecting in the brain, enclosed cavity, it could cause issues. Even in other areas, allogeneic material, there's a risk for immune rejection. There's difficulty in producing pure cell populations, and you want to have the cells that you're injecting as pure as possible. There's your cell processing, maybe requiring multiple steps. And when you're talking ES cells, there are obviously ethical controversies. Interestingly, as you all know, we've been developing iPS cells. So induced pluripotent stem cells have been a solution to embryonic stem cells, although not exactly replicating this activity. They do present most of the efforts and actually are going around a lot of the obstacles that are just mentioned. And when you're developing a cell product, you need to think of both the advantages as well as the disadvantages and how you're going to be addressing each of these issues. If we're going to adult stem cells, well, obviously there are a number of advantages. It's easy to get the cells and to isolate them. If we're talking autologous, then there's no rejection. You can use partially differentiated cells or induce the differentiation, and there are obviously less ethical issues. But there are disadvantages because the stem cells or the progenitor cells are usually rare in the different tissues. It might be difficult to isolate them and to obtain them in sufficiently large quantities that you are able to start a clinical trial. You may have poor proliferation capacity of these cells. There is limited longevity. These are not as sustained as ES cells. And in terms of multipotency, clearly they are less flexible, less differentiation potential, and there's a limitation to the use of autologous cells in that if they have genetic defects, then you might be replicating the defects. So you might be thinking of other sources, either allogeneic or genetically modified cells. So with that, there are a number of challenges when we're manufacturing a cell product. And one is obviously the first one, the most important one, I would say, is the scientific obstacle. And then you want to take that to the clinical stage, and therefore you want some clinicians with you to be working on developing your product. There are technical issues, and there are regulatory issues, and we're concerned with all of them, and we have to address all of them and find solutions so that we're able to fulfill the requirements for a clinical trial application. So if we're thinking of what's the conventional cell therapy workflow, we go from tissue processing, isolating the cells, and here you have a list of tissues that could be used, either bone marrow or leukapheresis from peripheral blood, or it could be a biopsy specimens, and you could be isolating adult stem cells. For that, you will need to select your cells, whether they're hematopoietic stem cells, and you may want to use antibodies and magnetic beads, for which you need equipment that is authorized, obviously. And if it's not authorized, you can still use them, but you have to define the appropriate qualification to ensure that they're going to do the work that you need them to work. You might want to add treatments. And here we've been working, for example, at photosensitizers. So actually you have an image here of a photosensitizer that we've developed that was ISO approved. And there are a number of other equipments that all of you might want to develop. And for each of these, if they're a device, then you have to think of what is the regulation in terms of device. although it may not be necessary to comply with all the device strategy, because what you're doing is you're injecting cells. So while this is an obstacle, and clearly you have to have a device that's appropriate, you may not have to file for a complete device regulation depending on the nature of the product. I'm emphasizing this because a number of people think that you have to have a specifically authorized device to do that. And because you're injecting cells, what's important is the quality of the cell and not obviously the quality of the device, but what you're doing is not selling a device. What you're doing is using a device to get your cells. So keep that in your mind as you're developing your strategy, and this could be discussed with regulatory authorities, for example. But you should not think of that as a necessary obstacle. And then you need to formulate your cells. How are you going to administer them? Are they going to be resuspended in a specific or buffer, a saline buffer? Are you going to be freezing the cells? In which instance you need to expose them to freezing medium. Is that medium appropriate? And then are you store them at 4 degrees? And if you store them, how long are you going to be keeping them? So you need to have information on the stability. And even if you cryopreserve, you have to demonstrate that the cells are stable at different time points. If you're using ES cells, actually this is a similar schema, but just to again emphasize the point that in the manipulation, you need to have tests to show the regulatory authorities that you have control over your process, that you've been evaluating the different characteristics of these cells, and that you know exactly what you're doing in terms of expansion, differentiation, sorting, and the characterization of the final product, which is really crucial, and how you're going to freeze and store these cells, or store these cells. And then when we're using MSCs, which is a popular source of cells for treating graft-versus-host disease, for example, but also coming of age for treating a number of other immune processes, I think there's similar activities that have to go through, and you need to be able to show that you have control, you know what you're doing at the different steps, and you're able to present regulatory authorities with data on the different steps of your process, and to ensure that you have a good high quality product. So in terms of regulatory framework, the whole idea, Health Canada is concerned, is protecting patients' right and safety. And clearly, while they have efficacy in mind, what your product clearly needs to be safe, and that's the first concern. So it has to undergo very rigorous regulatory framework control. And what you want to do is to prevent potential transmission of infectious diseases, and you may need to control for that. You need to ensure that you have clinical safety and effectiveness, and be able to monitor the adverse events in your studies, and you'll be able to, and that will be part of your clinical trial protocol. You can have a risk-based approach in that you don't necessarily need to control every element to the strictest precision, depending on what is the risk associated with each of the different steps of the manipulation, for example. And there can be some flexibility in terms of manufacturing, and we might be discussing some of that. Sorry. So in going through the chemistry, manufacturing, and control, which is an essential element of your clinical trial application, I indicated that this is really what they're looking for, is safety, especially if the product has never been tested in humans. And what you have to have control over the different elements. As I mentioned previously, raw materials, source controls. If you're banking cells, what's the source of your cells? How are you expanding them? How are you banking them? You have stability, you have process controls, and do you have appropriate characterization? And importantly, you need to have good assays at the preclinical level. But what you need to understand is as you're going from preclinical to phase 1, phase 2, and phase 3, the regulatory requirements are actually going up. So it's not necessary in a phase 1 to have everything in place in terms of full GMP. However, by the time that you get into phase 3, you must be full GMP compliant, and you must have product that's very well characterized. So as you're going along, it's tolerated that your tests are not necessarily perfect. But as you're going along, you need to improve on that and have full product characterization by the end. So you must be able to demonstrate that you have the capability to manufacture your product consistently. So in labs, we're able to repeat tests. If it doesn't work once, you repeat it and then usually get it to work. But then when you get it to work, you have to show that it's replicating and it's robust. Now that you've got it to work, it's going to work every time. So if you're manufacturing a product and it's working only 50% of the time, then you're not ready for clinical. You need to have something that's at least 90% of the time that you're going to be able to produce a product that's efficient. So, and you need to describe everything from donor screening, if you're taking cells from a specific donor or a pool of donors or a single donor that you're expanding the cell, what are the characteristics? Are these cells safe? Are they allogeneic or autologous? Why are you doing that? Have a good rationale for how you're doing this. What's your cell source? What is your mobilization protocol relevant? And is it validated? Do you have appropriate controls? Do you have experience in doing this? And experience in the collection of the cells and in the recovery? What's your recovery level? What is your procedure in order to obtain these cells? And we went through, like, if you need immunomagnetic separation, for example, what's the data on the recovery? What have you been obtaining in terms of results to do that? And probably one of the most important elements is the release criteria. The release of what's your starting material, like what's coming in, obvious, garbage in, garbage out. So do you want to manipulate cells? If you're starting with a product where you don't have enough cells, probably you don't want to go through the expense. So you need to have very good starting material release criteria and also release criteria in the end. Like what are you going to be giving to the patient? And you need to develop an analysis that are very reliable and set acceptance criteria that are well defined that will allow you to initiate the process in this case. You need to have a manufacturing process that is under control, so it needs to be consistent from one sample to the other. And you must have all the elements characterized, your starting material, your intermediate products, and your final products, and focus on acceptance and release criteria for all of them. When possible, you should have material that has been administered to humans and that is compliant with human administration. When it is not, you must have detailed information on your reagents, on your media, on your buffers, on your components, and have certificate of analysis, like the COAs that we always refer to. The certificate of analysis ensuring that you have the right product with the right qualifications, and you must be able to provide references of that product to the regulatory authorities, and this must be part of your clinical trial application. Process development and manufacturing controls. Well, clearly you must be starting with a small, well-established, small-scale procedure. That's what you're doing in your research lab. So you must have full control over this step, and what we want to do is actually scale up. Go from a small size into a size that will allow you to treat patients. So you need to be able to have your product and actually share with a team. The best thing is to go around and look at who has experience in these settings, who can help you set up scale up and use the right equipment to do that. Don't try to do that on your own. You're going to lose a lot of time. Go to people who have experience, get partners, and then develop them in association with them. Never drop your product, because as a discoverer, you're the ones who have most knowledge of how to do these things. And determine the start number of cells, the size of the raw material that you need. Are you able to get these in the right quantities? And are you going to be able to afford all the reagents? Because the antibodies, the growth factors, they can be pretty expensive. You need to optimize and standardize. Growing cells in a large volume is not the same as growing them in a Petri dish or in a 96 well plate. So you have to control for all these aspects that are listed here. And you must be able to characterize your process, your cells in process and in the final. And you need to have very good specifications that ensure that your product is safe and is of high quality, and that's why these items are in red. You need to be able to measure the integrity of your product and how stable it is. You need to have good product parameters to ensure that you're not generating adverse events. And if you're including elements, for example, growth factors that are associated with adverse events, then these are things to be looking for that you need to be pointing out in your clinical trial application. And you need to demonstrate control of the manufacturing product and ensure lot-to-lot consistency. And define the product release specifications. This is really crucial element. So in terms of safety assessment, I indicated already a number of elements that you need to include in your application. You have to have safety testing during the process scale-up. Consider safety testing for the qualification during your dry runs. Identity and identifying the right tests to be doing to ensure that your product is of high quality. And it should be as close as possible as the cells the tests that you have been developing in your lab. But think that you are going to be needing to do the test on the product that you're manufacturing on a real-time basis. So don't think of doing a long-term mouse assay that takes 4 months as your qualification test. You need something that you can do quickly that may not be as good as the one that you've been doing, but that is enabling you to do the right and rapid qualification. And you need in-process quality QC samples that you're going to collect and store so that you're able to go back and characterize your product. So in terms of, in the end, what you need is a product that has bioburden evaluation, so sterility testing, so 14-day microbiology culture, and obviously if you do that, you will need to freeze your cells. If you're doing fresh cells, you will need a surrogate, either a gram stain or a part-time or early, I would say intermediate microbiology culture, which might be better than a gram stain because there are lots of risk of false positive gram stain. You need pyrogenicity, so a luminous endotoxin assay that is commonly performed, that is within spec. You need mycoplasma testing, and you usually need to ensure that the source of cells are not infectious, and therefore test for these various agents before producing. For CAGJD, it's a screening, not a test that you are performing, but the others are usually tests that you are performing on the cells or on the patient that you are using as a source of cells. In terms of the cells, you need to ensure that the cells are as pure as possible, not necessarily 100%, but what you need is that you have control over that and that you have a high quality product. If needed, you need HLA typing. You can have, if you're deriving your cells into cancer cells for vaccine purposes, You need to have control over that. If there's possibility of introducing cancer cells, if you're along with your autologous cells, you have to look into this, introduce elements that eliminate or decrease the possibility of causing issues. And definitely you need viability and integrity of the cells, and you need to have some marker and measurements of viability, recovery, and release of the various components, including people will be asking, what are the other cells that you're infusing? What are the impurities? And in terms of safety assessment, and this is usually collected in your preclinical data. How is the homing? How is it going to be occurring? Are the cells going to go to the right area? Do you have data to support that, if possible? Not necessarily with every type of experiment, because there might be already data with administration of these cells. And historical safety data is very important. I think if you can include that, that's clearly going to help your application. And you need to adapt your product and have risk assessment. I think it's clear that depending on the nature of the drug, the disease that you're treating, and if you're treating end-stage disease with no therapeutic option, you're going to be allowed much more leeway than if you're addressing a condition for which there are already treatments available out there. And you need to obviously provide the regulator with as many reliable preclinical data. And I would say the best thing is to be confident about your process. If you're an investigator, you've been doing the studies, you know how it works, you know what's the best timing to go into the clinical trial when you know how it works. So in terms of safety, I already mentioned the importance of purity and identifying the nature of the cells that are in your product that you're administrating, along with the therapeutic cells. You need to ensure that these are not causing genetic defects. Might not be pertinent to what you're doing, but this will have to be included in your application. Karyotypic stability and generation, whether you're generating teratoma, especially if you're using ES cells or iPS cells, how are you going to be addressing this possibility? Identifying cells, there are a number of ways. Don't want to go over them, but just to say that number and viability can be done using a variety of measurements, and you're in control of doing that. You have to justify what type of instrument, what type of methodology you're using, and show that you have control over that. This can be in different flavors, and you can have morphology, not only the measurement of the number, but also the quality of the cells. And you will have to have some potency assay, show that the cells are not only there, they're not only biologically have biologic markers, but you have indicators that these cells are potentially active. So you need to define the right potency assay. And if something, I would say that's probably the most difficult thing that you need to integrate. And a potency assay that ideally you can generate within a short time as possible so that you can release your product for patient administration. So, and for potency assay, you have functional assays. And I indicated already that in a phase 1 study, you might have a functional assay that's maybe not as well sophisticated as what you would do for a phase 3, but provides you with sufficient information for doing the trial. There are surrogate assays that could also be acceptable, and then that's something that you may want to discuss with Health Canada, and you will develop these as you're going along to your phase 3. And you need to demonstrate that your product is stable, and therefore that you're starting cells, how long can you keep the cells before starting to treat them. So you need to identify your shelf life of the initial product and also of the final product. And that's the same whether it's fresh or cryopreserved. You need to demonstrate that your product is stable for the use that you're going to take advantage of. And finally, in the testing procedure, you need to be confident that these cells are reliable and that they are robust. These are the criteria that you will use to say, yes, I'm giving this patient to the patient. So if it's a personalized therapy, you'll have to fill out release criteria for every individual sample. You're going to be monitoring your manufacturing process, the stability, and incorporate process changes and inform Health Canada if you change anything while you're doing your trial. And all the instruments that you are using, you must show that they're installed properly, that you have operating qualification and process qualification, that you have a clear maintenance plan for all this instrumentation, that they're well calibrated, and they have the right procedures to operate them. And the same for the analytical methods. So with that, I would like to end this presentation and basically thank the people that have been really key in devising these strategies at our center and that are always introducing new aspects, our directions, our operation direction, the quality assurance team, the various production teams, the gene therapy team, the quality control team, the translational team, and our research team that are coming up with the great ideas that we're putting into patients. With this, we're often partnering with C3I, which is providing us with also infrastructure to help put together a clinical trial and develop that further. So I want to thank all of them. I also want to thank our clinical teams that are heavily involved in designing these various trials. And we have people all across Canada that are involved in the different centers. And actually, there's people from every center that are involved in various clinical trials. So I thank all of them. Special thank to Christian Scotto, which has been extremely important in setting up actually this presentation and has been really crucial and has written a lot of what I've been presenting today. Martin Giroux, similarly, that's been inputting and is heading our manufacturing center. They're my research lab, and obviously all the funding organizations that you can see here that is partnering, Stem Cell Network, BioCanRx, T-Cell, C3I, and others, and Genome, and all the others. So with that, I would like to let you go over some of the collaborations that CellCan is having with a number of networks and the coming activities before we open up for questions.
Oh, yes, so we have a few activities upcoming, November and December. Take a look at them, see if you're interested in joining. We've got the Journée annuelle du réseau T-Cell. We've also got an ISO meeting with RMAC in Toronto. And then in collaboration with CCRM, we're holding a logistics and supply chain workshop. And that's coming up in January. We also have a continuation of this lunch webinar series. So thank you everyone for taking the time to join. I hope you found it helpful. I know I sure did. And I would also like to thank Dr. Denis Claude for giving us all of this insight based on his tremendous amount of experience in the field. So for everyone who's listening, if you could just throw your questions in the chat box. Chat box is on the right side of your screen. We also have a survey for you to take at the end, which I will send out to everyone so you can see where the chat box is. And I will get this started with some questions. So I had a quick question. You mentioned that there was a lot of, you mentioned that you were interested in asking for help with scale up and that you didn't recommend that people try to do that themselves. Could you give us an example of where you went to look for resources to help for scale up of processes?
Yeah, actually, I think that, for example, in our case, we went to our cell therapy manufacturing lab where we've developed actually a translational platform. And these are individuals who have research lab experience that actually have been exposed to what's occurring in a clean room environment and clinical setting. And therefore, they have knowledge of both areas. And what they're doing is they're very familiar with the types of tubings, the types of materials, the flow cytometry, measurements, and qualifications that are needed. So therefore, they are able to take our research personnel, take them through, go over the process that they are doing, and think of how to implement that and how to get this together so that we end up with a cell product that we can give to patients. And while we have lots of experience with manipulating T cells, there are other individuals that have various experience in various tissues.
Right.
So I think that you should come to CellCan, for example, for advice, and we can probably orient you towards the centers that have the right experience. For example, Ottawa has been working a lot on mesenchymal stem cells. People in Calgary have been working in Edmonton, and Calgary also, but mainly Edmonton on islet cells. Greg Corbett, Gail Pia have been working very hard on getting their GMP together, and they have resources to help people in this environment. People in Vancouver have other protocols, people in Toronto. So every city, basically, Hamilton, there are people with various expertises, and we can help you if you don't know the right people. I would suggest that you send a quick message to CellCan and to various people, and send us an email, touch on on the email address, and send us an email, and we'll contact you and maybe try to find out which is the best resource for doing this.
That's great. Thank you for the offer. And also, we have some questions coming in on the chat. So in general, what is the efficiency of differentiation of ESCs or iPSCs? Do you have a general?
I would say it depends on the nature of the cells. Obviously, if you want to get hematopoietic cells, I'd say, and it's also on your reagent. For example, we have a protocol right now that is generating, expanding hematopoietic stem cells out of hematopoietic stem cells. So not ES or IPS, but actually from hematopoietic cells, and within 5 days we're able to generate a multiple, like a more than 50-fold expansion on the cells in usual conditions. For other protocols, it's highly variable. I know, for example, in skin, you might want to start with a few skin cells and you end up with whole tissue, but it could take a couple of months to get that. So it all depends on the nature of the cell and the type of end product and how differentiated you want your cells to be. Also, maybe I want to emphasize that you don't necessarily always want a highly differentiated cells. It may be when you put them in the right environment, they're going to continue to differentiate. And the reason is that you don't want to exhaust your cells in doing so. I'm sorry, I'm being a little general here, but it really depends on the nature of the tissue and of the starting material.
Okay, great. And another slightly related question. You mentioned there were different types of inducible cells for therapeutic usage. Are there any advantages of one cell type over another for long-term tracking?
If you have cells that have depending on the source of cells, if you are using allogeneic, you could track them using, for example, HLA or other characteristics you could do. Even if the cells are not expressing that, you could find ways of tracking the cells, identifying a molecular marker that you can use to identify the cell, especially if it's allogeneic. In the autologous setting, if you're modifying the cells genetically, you can track down your genetic modifier.
Right.
And it's not as much the regulatory authorities that are going to ask you about monitoring the homing of the cells, although it's important that you have demonstrated that they are able to generate the activity that you want and that they're going to right tissues. But it will be important to determine the efficacy of your product. If you want to take your product further, people are going to ask, well, are your cells still there? Is the effect that you're seeing due to the cells that are present? And you will want to show that your cells are sustainable. And for example, if we're talking CAR T cells, you will want, it's been demonstrated that if the cells are surviving, you have greater activity. More sustained activity goes along with persistence of the cells in many settings. So you will want to demonstrate that. So I think it's a good argument in terms of generating a product that you're going to be able to take to the next level as much as possible.
Another question, what happens if the starting material quality is poor, but you're limited with the availability of the starting material?
Yeah, that's a great question, and that's something we encounter. Sometimes we start with very small numbers of cells and you have to expand them. So I would suggest, if you have do you have control on the initial input of cells? Like, can you increase, and can you add a selection process? Can you initially do like not a full selection, but a pre-selection on precursor populations that could increase your end product? And I have an example where recently for T cells, if we selected specific subsets initially, we could increase the end product. So that's a way of doing that. The second way is to actually improve on your expansion, which is not easy, and you can either increase the number of growth factors, you can increase the duration of the incubation, or actually, and something that people have not been looking at very much is improving the conditions. So maybe adding some matrices, matrix, where you can have some binding with cell surface. Sorry. Am I doing something wrong here?
It's fine. For the sharing.
Sorry about that.
So you were mentioning the matrices?
Yeah, I was mentioning the matrix actually can help grow cells quite efficiently. So it's something that is only starting to be investigated. So I would say you have to look into this.
Okay. Interesting.
Or at least that you don't have to, but that's an option. And maybe the other thing is automation. A lot of people wonder when to automate or how to automate. I think if useful, you should automate. And but you don't have to do this early. I think usually automation is causing, it could cause a delay. And you're better to start your trial and demonstrate that you have a proof of principle while at the same time to work on automation if you can. But if not, I've seen products go from phase 1 to phase 3 without any automation, with a hand-on process. Then it's better. It's better to go quickly into the clinic and generate data than wait to have an optimized product, or at least that's my my take on that question.
Great. And another question from the chat. You mentioned there was about purity of cell-based products at release. So what's an acceptable percentage of the desired population of those cells at the release?
Say that again, can you?
So you were talking a little bit about making sure that the purity of your final product was good. Do you have a number value of what's the percentage of cells that you would want of your desired cells?
Like ideally, it would be, I would say ideally it's more than 90%.
Okay.
But it's not a fixed number. Like people think that, oh, I have to have always more than 90%. I think we've gone with a very low percentage of positive cells. For example, we could go less, if you're able to justify that you have very low numbers of cells, but they are present in your product, I think that then it's possible to go ahead. And I would say probably more than 50% is acceptable. And sometimes even much lower than that. And I've seen protocol where it's much lower, but then the nature of the contaminating cells are very important. You need to demonstrate that could these cells be administered. So for example, if you're talking stem cell transplantation, are these cells normally given in stem cell transplant? So then you could have a rationale for saying, well, my contaminants are normally used. So there's no deleterious effect. If they were used in the same setting, I would not cause issues. And you're counting that your cells are going to be expanded in vivo.
Okay, so if anyone has any other questions, put them in the chat now. Other than that, we've got another question. So what is the long-term and short-term future perspectives of cell therapy, considering the coming of 3D cell printing?
Yeah, that's a great question. Actually, I was discussing that recently with people that are thinking the solid organ transplanters actually are seeing this coming, and they're seeing cell therapy. They were seeing cell therapy as actually how do you say, a competitor to organ transplant. And interestingly, they are seeing 3D printing as a way of prolonging their half-life. Now, I am teasing here, but actually there's lots of potential. The solid organ transplanters are seeing that there is benefit. For example, there's groups that are decellularizing organs and then generating organs. So actually, sometimes we can't administer cells. Cells won't be doing the job by itself. You need to get the whole organ. And therefore, 3D printing is another option to generate organs. Clearly, some organs are easier to replicate than others. Probably liver is less complicated than kidney, but nothing is impossible. And people have great imagination, and please continue doing the great work that you are doing. This is fascinating, and we need to be taking your findings into clinical application. And the 3D printer is actually, there are clinical trial application compliant machines that can be used, and depending on the source of cells, you could probably generate organs in a very near future that will be possible to transplant. So there's much potential for both cells and tissue and organs, And we haven't even talked about gene therapy, like gene modifications, insertion of genes into humans. That's another potential, but easily accessible is the administration of cells that have been genetically modified. And this we were touching upon, and we haven't gone to the details, but this is another area that obviously people are, all the research community is highly interested in.
Great. Well, thanks once again, everyone, for joining. We're just about out of time now. Thank you, Dr. Denis Claude, for giving us this presentation. And we'll be having these again every month. So check out our website, fill out this survey at SurveyMonkey so we can get a bit of an idea of what you guys think of our webinars. And we will see you next time.
We need your feedback. Thanks very much. Thanks to all of you for attending.